My name is Kylie Stazeski, and today is October 23rd of 2024, and this is my presentation on gene therapy. The article Gene Therapy Comes of Age by Cynthia Dunbar, Catherine High, and Michael Sustain portrays gene therapy as a promising treatment for both inherited and acquired human diseases such as hemophilia, ocular and neurodegenerative disordered, and lymphoid cancers. The article summarizes the milestones of development of gene therapies as well as discussing the in vivo and ex vivo administration of viral vectors and the adoptive transfer of genetically engineered T cells. The article also discusses the further advancements of genome editing and technologies. The background information in this article portrayed the original hardships of gene therapy. Theodore Friedman was one of the first researchers to provide a prophetic account of the foreseeable challenges of gene therapy to treat inherited monogenic disorders. At the time, mass interest in gene therapy was sparked by the potential that single treatments could offer potentially curative results. Researchers hypothesized that in contrast to protein-based drugs that required repeat treatments, gene-based therapies could offer and sustain production of endogenous proteins, for example, the clotting factors that would affect people diagnosed with hemophilia. There was continued translation of gene therapy-based concepts to patient care, which began in the early 1990s. Unfortunately, the clinical trials at the time proved to have disappointing results, as the gene therapies at the time provided no clinical benefit. In fact, no real benefit of gene therapies have been seen until at least the last 10 years. There are currently several gene and gene-modified therapies and approved drugs being used around the world as of now. The authors then went on and discussed the following as evidence. Recombinant replication defective viral vectors were the first molecular tool that enabled the efficient and non-toxic gene transfer into human somatic cells. Retroviruses and adeno-associated viruses have shown the most clinical promise. Retroviruses facilitated the production of vectors capable of undergoing reverse transcription, all while lacking the ability to replicate in the human body. In the 1990s, Y retroviral vectors were first used to deliver genes to populating HCCs. In current times, lentiviral vectors are typically used to aid gene therapies as they can carry larger and more compact gene cassettes. Another advantage of lentiviruses is that they are able to integrate into the coding regions of genes. They are currently the tool of choice for most HSC applications. Adeno-associated viral vectors, also known as AAVs, are engineered for non-pathogenic and non-developed parvovirus that is naturally defective in replication. The viral coding sequences of AAVs are able to be replaced with gene expression cassettes. One limitation to these AAVs is that they can only carry a small amount of DNA when compared to Y retroviruses and lentiviruses. In comparison to viral vectors who can only mediate a single type of gene modification, genome editing technologies can edit the addition of genes, gene correction, and other modifications. Genome editing can be performed cells in vivo or ex vivo. Targeting organs in vivo avoids both practical and regulatory landmarks of ex vivo gene delivery. In vivo approaches are highly dependent upon the tissue of target. In contrast, ex vivo delivery is derived from HSCs, which of course produces ethical hurdles. Gene editing typically involved a targeted DNA alteration, which is first initiated by the creation of nucleus-induced double-stranded break, which stimulates the recombination of mammalian cells. Non-homologous end joining mediated repair results in the creation of variable length insertion or deletion, which generally inactivated gene function. Homologically directed repair can be used to create specific alterations to gene sequences, which following recombination results in correction. Finally, chimeric antigen receptors, which were discussed as promising tools in gene therapy. Chimeric antigen receptors, also known as CARs, are synthetically engineered receptors for antigens and, with a single molecule, are capable of reprogramming the specificity, function, and metabolism of T lymphocytes. CARs can be engineered to reconcise proteins and other molecules, thus creating an exceptional treatment. On a personal level, I feel that this article does a tremendous job of illustrating the history of gene therapy as well as its relevance and its prospective outcomes. Specifically, the authors discuss the key differences and the pros and cons between the use of retroviruses, lentiviruses, and pure gene editing. They created a virtual Venn diagram almost that made it really comprehensible. In addition, the article spotlighted the highs and lows of the journey of gene therapy, which really allowed me as the reader to learn without developing a biased opinion. That's it. Thank you for watching.